A very good morning to everyone. Welcome in the class. As we say in the starting, let's crack CSIR UGC NET and GATE to 2021 together. We're watching this platform under the umbrella of Unacademy. Unacademy is one of the India's largest learning platform for the preparation of CSIR UGC NET and other competitive examination. I'm your educator Farida Johar Bandukwala. I have a teaching experience of 10 years. I've qualified MP SLET examination in year 2018. If you're opting for Unacademy, what all things you are going to get? You are going to get daily live classes. Live classes is going to keep you into that momentum of studies every time, and live classes is going to give you that freedom that you can ask your doubts and queries while the class is going on live. Then the whole syllabus is quite structured. As you know that for any competitive examination, the syllabus is quite lengthy. So being syllabus in a quite structured pattern, you will be able to complete it in a small span of time. The live tests and quizzes are taken so that you can evaluate yourself that how much you have learned so far. And then you will get unlimited access towards all the videos and all the things which are recorded in this particular category. So free live classes are conducted on two platforms. One of the platform is YouTube and another platform is special classes. So you're watching this video on YouTube. So what happens in YouTube, you just go to the channel, subscribe the channel, and you will have the access for all the videos which are going on live or the videos which were previously live. Then for special classes, you need to download the Unacademy Learning app. After downloading the app, you will be asked to make your account. After making account, you can use the referral code of an educator. That is in my case, you use the referral code FB01. If you will use my referral code, you will be able to watch the free live classes of mine as well as other educators. After that, there is this plus subscription. This plus subscription is a paid subscription, and do not worry seeing the paid term, as there is a large discount going on. For 12 months, it is going to cost you just 11,550 rupees, and for 24 months, it is going to cost you 16,800 rupees. And what my suggestion is that these two are the best best packages available, and which is going to help you further. And further, you can use my referral code, again the same referral code FB01, for getting an additional 10% discount in the plus subscription as well. So this plus subscription will be costing you 10,395 for 12 months and 15,120 rupees for 24 months after using my referral code. Now the topic of today is protein sequencing strategies or protein sequencing. Protein sequencing. So we are going to talk about this. We have previously seen that how the DNA is sequenced, and now we are going to see that how protein is going to be sequenced. So what is protein sequencing? Protein sequencing is a practical process of determining the amino acid sequence of all or a part of protein or a polypeptide. We know that a polypeptide is made up of large number of amino acids joined together. This is the sequence of a polypeptide or a protein. So we want to know that which one is the amino acid number 1, which one is the amino acid number 2, which one is the third one, which one is the last one. So all these things can be easily determined by protein sequencing. So in year 1953, there was a scientist. His name is Frederick Sanger. Frederick Sanger. He belonged to Cambridge University in England. He reported that the amino acid sequences of two polypeptide chain composing there are two polypeptide chains in the protein, that is insulin. That was the first idea, that a protein is made up, can be made up of more than one polypeptide. So insulin is made up of two polypeptide chains. This was told by a scientist, Frederick Sanger. So Sanger's result was clearly established that all the molecules of a given protein, they have a fixed amino acid composition, a defined amino acid sequence, because if there is difference in the amino acid, then only there will be difference in their functioning. And therefore an invariant molecular weight In short, proteins are well defined chemically. So Frederick Sanger, he told that insulin is made up of two polypeptides, and then further it was established that each protein is different from each other. Each protein is different from each other. Now we know this, right? But when the whole story started, this small thing was a big idea for that particular time. So each protein is different from other in terms of two things, in terms of amino acid sequences and in terms of molecular weight. So if amino acids are different, obviously the molecular weight is going to be different. Then this scientist in year 1958, he was awarded Nobel Prize for his contribution to determine the structure of insulin and other proteins. So Nobel Prize for insulin and other proteins structure. Insulin and other proteins structure. So an alternative approach was also given by another scientist, and his name is Per Edman. So Edman in year 1950, he gave a different approach, and this allowed the determination of the extended sequences of the peptides or the whole protein sequence, and which is widely used till now. So Edman sequencing is very famous and it is used till date, right? And he gave this idea in year 1950. Now talking about protein sequencing strategies, we'll be talking about two of the methods, that is Sanger's and Edman. But before that, let us talk about the protein sequencing strategy, that what will be the basic strategy, whatever technique you take, right? So we'll be seeing this. So the usual strategy for determining the amino acid sequence of any of the proteins or whichever method you are going for, it is made up of, or it is consisting of 8 basic steps. 8 basic steps. Now, what are these 8 basic steps? If a protein contains more than one polypeptide chain, the chains can be separated and purified. One thing. If protein contains more than one polypeptide, then each chain needs to be separated. Then each chain needs to be separated. One thing. Then intra-chain bonds, just like sulfide bonds, disulfide bonds, then cross bridges between the cysteine residues in the polypeptide, they need to be cleaved. If they are not cleaved, then the result cannot be clear. So if disulfide linkages are there, then the disulfide linkages should be broken down. So next is intra-chain linkages should be broken down. Intra-chain linkages should be broken down. Intra-chain linkages should be broken down. Then the amino acid composition of each polypeptide chain is determined. Amino acid sequence of each polypeptide is determined. Then if we are going for the determination of the sequence, then we would like to know that what was the amino acid sequence at the N terminal and what was the amino acid sequence at the C terminal. So N terminal and C terminal residues are determined. N terminal and C terminal residues are identified. Right. Then each polypeptide chain is cleaved into smaller and smaller fragments so that we can evaluate each and every fragment. So polypeptide is broken down. Polypeptide is broken down into small segments. To small segments. Then after this, sequence determination of peptide will be done. Sequence determination of peptides. Sequence determination of peptides. Then the overall amino acid sequence of the protein is reconstructed from the sequence in the overlapping fragments. So overall amino acid sequence is reconstructed. After that, the position of the cross bridges, that is sulfide bonds, will be determined. Position of intra-chain linkages is determined. After that, we are going to talk about the N-terminal amino acid analysis. So, next is N-terminal amino acid analysis. N-terminal amino acid analysis. So, we know that amino acid chain has got one of the N-terminal and one of the C-terminal. N-terminal, why it is called as N-terminal? Because it has a free amine group, and C-terminal, why it is called as C-terminal? Because it has got a carboxylic acid free group at the C-terminal. So, a generalized method for N-terminal amino acid determination would be react the peptide with a reagent that will selectively label just the N-terminal. If there is one thing which is going to react with only N-terminal and it is going to label the N-terminal, then it is possible to know that what was present at the N-terminal. So, selective reagent which binds with only N-terminal amino acid, N-terminal amino acid. After that, second is hydrolyze the protein. The protein which is to be analyzed, it should be hydrolyzed so that now it can be determined. Just the binding was the first step. Second step is to hydrolyze the protein. Hydrolyze the protein. After that, determine the amino acid by chromatography and comparison with the standards. So, chromatography is performed. So, by chromatography, it will be compared with the standard so that it can be identified that which amino acid was there. So, determining which amino acid forms the N-terminal of a peptide is very important and useful in identifying the order of individual peptide fragments, right? And the sequence of whole chain can be analyzed by this. Because the first round of Adam's degradation, which we are going to see further, is often contaminated by impurities. Often it is related to the contaminations, and therefore many of the cases it does not give the accurate determination of the N-terminal. This is the problem with Edman sequencing. Now, second thing that we are going to see the general strategy for C-terminal determination. C-terminal amino acid analysis. This will be a better term to be used. C-terminal amino acid analysis. So the number of methods which are available for C-terminal amino acid analysis is much smaller than the available techniques for the N-terminal. N-terminal tends to be more reactive as compared to C-terminal. So number of methods for C-terminal determination is quite larger as compared to the N-terminal determination. After that, the most of the common method is to add the carboxypeptidase to a solution of the protein, then take the sample at regular intervals to determine the terminal amino acid by analyzing a plot of amino acid concentration against the time. So the most common strategy is use of carboxypeptidase. Use of carboxypeptidase. And then further determination, right? So this method, use of carboxypeptidase and regular sampling to identify the amino acid. To identify the amino acid. So this method will be very useful in case of polypeptides and protein which are blocked at the N termini. Those which are actually joined by the N termini, or somehow they are blocked or they cannot be retrieved in a proper way. So for them, this technique is quite helpful. So C-terminal sequencing would greatly help in verifying the primary structure of protein predicted from the DNA sequence, and to detect any kind of post-translational modification which is occurring after the protein has been formed. So useful in determining useful in determining primary structure of the polypeptide, right? So this is the C-terminal method, generalized. Now, as I told you that we are going to talk about Sanger's method and Edmonds method in detail, so we are going to talk about Sanger's method. Sanger's method. So this method was given by scientist Frederick Sanger. He had demonstrated a method to determine the amino acid residues which are located on the N termini end of the polypeptide chain, right? So a method which determines a method which determines the amino acid sequence which is located at N termini. Excuse me, can you just close the door and put the latch? Thank you. Sorry for the disturbance. A method which determines the amino acid sequence which is located at the N termini by the use of a reagent which is called as FDNP, which is done by a reagent FDNP. So what is the full form of this reagent? It is fluorodinitrobenzene. Fluorodinitrobenzene. This is the name of the reagent which is used. So Sanger showed for the first time that amino acids are covalently linked together by the amino acid and alpha carboxy group. Very important that he told for the first time that they are covalently linked by the alpha amino and alpha carboxy groups. Then he suggested the stepwise release and identification of amino acid starting from the N termini. So I would put this N termini into this thing, so that the thing that will click to your mind whenever you are talking about Sanger's method, it is N termini. Then he used the reagent that is fluorodinitrobenzene, which is commonly called as Sanger's reagent. So whenever you come across the thing, what is a Sanger's reagent? So Sanger's reagent is fluorodinitrobenzene. So FDNB reacts with the free amino group of the N termini, and upon hydrolysis a yellowish colored compound or a derivative is produced called as dinitrophenol, and that is how the N terminal is determined. So this is called as Sanger's reagent. So this FDNB reacts with free amine group, free amine group of N terminus. After that, upon hydrolysis, a yellow colored derivative of N-terminal amino acid is produced, which is called as dinitrophenol. A yellow colored derivative dinitrophenol. Dinitrophenol is produced. Is produced. So this was the basic. Now the dinitrophenol amino acid which is produced is identified by comparing it with the known standard dinitrophenyl amino acid complex. by using gel electrophoresis, right? So dinitrophenol amino acid is compared with dinitrophenol amino acid standard one through gel electrophoresis through uh, it's not electrophoresis, it's chromatography, gel chromatography, gel chromatography. Why it was thought by many of them that that at most this method can tell and provide the sequence of the N terminal, but when the Sanger's method was taken a step further by using several proteolytic enzymes such as proteases, then chymotrypsin, then trypsin, and partial hydrolysis, an early version of chromatography, Sanger was able to cleave the protein into small fragments, and when these small fragments were fit together like solving a jigsaw puzzle, it gave the whole sequence of the protein. So if you come across a question that the uh, Sanger's method is capable of identifying the whole sequence or not, so the answer would be yes. By using several other enzymes such as the proteolytic enzyme, the Sanger's method can be even used for determining the whole sequence. So till year 1955, Sanger was able to present the complete sequence of insulin, and that is why he was given Nobel in year 1958. So Sanger's method clubbed with various proteolytic enzymes gave full sequence of proteins, and that is why he was able to give the whole sequence of insulin till year 1955. So the peptide, which is the small peptides which is obtained, they are hydrolyzed into the constituent amino acid by what process? By heating it with the hydrochloric acid at a very high temperature, that is 110 degrees Celsius, and they are incubated for 24 hours. So the peptide is hydrolyzed by treating with hydrochloric acid at 110 degrees Celsius for 24 hours. This is the step of hydrolysis. So let us have a idea that how it looks like. Let us draw dinitrochlorobenzene. So it looks like this. It looks like this. The dinitrochlorobenzene. It reacts with the polypeptide. So polypeptide, let us draw a polypeptide also. Here it is having a free amino group, and here it is further having a long chain. So it is going to react with it. This is going to be released out, and we are going to get a yellow colored compound, that is dinitrophenol, which is going to look like this. See, this is how we are going to get. So upon hydrolysis, the N-terminal amino acid, it has got, when this is hydrolyzed, it has got this chromophore. This is the chromophore attached, and which can be separated and which can be identified. So this is how the process of Sanger's process occurs. When it is cleaved out at this place, this is going to give certain kind of color, and then it can be determined. So this was the process of Sanger's method. Now let us talk about the Edman degradation. Edman degradation. So admin degradation is a very important process for protein sequencing because it allows the ordered amino acid sequence determination. So there won't be any kind of jigsaw puzzle which we are going to solve, but it's a sequential method and very easily it will be performed. So allows ordered ordered amino acid composition of a protein which is to be discovered. After that, there are various automated sequences which are based on the principle of Edman degradation, and they are widely used and are able to sequence the protein up to 50 amino acid long. So automated Edman sequencers are also available. Automated Edman sequencers are also available. After that, the Edmond degradation method, it is based on the principle that the single amino acid residue can be modified chemically such that it can be cleaved from the chain without disrupting the bonds between any other residues. This is the thing that Edmond, it is based on the thing that amino acid residues can be modified chemically and they can be cleaved from the whole chain without disrupting the bond between the other residue. It is based on this principle. So the procedure can be achieved, or the procedure it is performed with a very minute amount of peptide. We don't need a large amount of protein sample also in Edmond degradation. So small sample is needed. Not large sample, small sample is needed. Usually the amount when we are saying small sample, it is so small that it is 10 to 100 picomoles. If you want a very successful completion, so lesser the amount, lesser the mistakes, and more is the uh, resolved sequence of the amino acid which will be obtained. So I hope you people are following till now. I hope you are understanding. So write in the chat box if you are able to understand the things. Show some response, yes or no. Now talking about the steps of Edmond reagent. So in Edmond degradation, or in Edmond's reaction, or in Edmond sequencing method, what all steps are there? First step is break any kind of disulfide bridges which is protein present in the protein with the use of a reagent which is called as 2-mercaptoethanol. So step number 1 is break any disulfide disulfide bridge by reducing agent by reducing agent, and the name of the agent is 2-mercaptoethanol. 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 So a protecting group such as iodoacetic acid may be necessary to prevent the bonds from reforming. So when we are just breaking the bond, we don't want them to reform the bond. So to stop it from reforming the bond, iodoacetic acid is added to prevent the bonds from reforming. Iodoacetic acid is used, right? Iodoacetic acid is used. The first thing that we have seen over here is mercaptoethanol, which is responsible for breakdown of the bond, and to make sure that the bonds are not reforming, iodoacetic acid is used. Then the next step is to separate and purify the individual chains of the protein complex if there are more than one chain. So separate and purify individual chains. Individual chains. Then after that, determine the amino acid composition of each chain. Determination of amino acid sequence. So break each chain into fragments under 50 amino acid long, not more than 50 amino acid. So the sequence which we have got, they should be broken down into fragments of 50 amino acid long. Then separate and purify the fragments. Separate and purify the fragments. After that, determine the sequence of each of the fragment. Repeat the different cleavage and determination, and then there will be construction of the whole sequence. So this is the basic. Now we are going to start with the Edmonds reagent, Edmonds sequencing. So first step is digestion into peptide fragment. Digestion into peptide fragment. Digestion into peptide fragments. So digestion is done either by endopeptidases such as trypsin and pepsin, or by chemical reagents such as cyanogen bromide. So different enzymes are going to give different cleavage pattern, we know that, and the overlap between the fragments can be used to construct the overall sequence, because different type of enzymes, different type of chemical reagents, they are going to give, or they are going to cut the whole sequence at different places. So overlapping things will be used for constructing the whole sequence. So digestion is done by either endopeptidases, and these endopeptidases could be trypsin or pepsin, or it can be done through chemical reagents, and the chemical reagents which could be used are cyanogen bromide, right? So this is the digestion. After that, the step which will take place, that is the main step, that is the reaction step. So the peptide which is to be sequenced, it needs to be absorbed on a solid surface. The sequence which is to be determined, it needs to be absorbed on a solid surface. So one common solid substrate which is used is a glass fiber which is coated with polybrine, which is a cationic polymer. So we'll just make this crude picture. So this is the glass fiber. This is the glass fiber. Now this glass fiber is coated with a cationic polymer, and the name of this cationic polymer is polybrine. This is polybrine, which is a cationic polymer. After that, there will be this polypeptide chain which will be attached to it. So the Edmond reagent, what is the Edmond reagent? Edmond reagent is PITC. PITC. What is the full form of PITC? It is phenyl isothiocyanate. Phenyl isothiocyanate. This is the Edmond reagent. So after this thing is formed, this entity is formed, that glass fiber is there, polybrine is there, and the amino acid sequence is attached to it. Phenyl isothiocyanate will be added to the adsorbed polypeptide together with mildly basic buffer. So mildly basic buffer solution which is used in this case is trimethylamine. Trimethylamine. Oh, trimethylamine. So this is going to react. This is sorry, there was some technical issue. So trimethylamine is added. This reacts with the amine group of the N-terminal amino acid. This phenyl isothiocyanate is going to react with the N-terminal of the N-terminal of the amine group. So the terminal amino acid can be selectively detached by addition of the anhydrous acid, which contains no amount of water. So see, if this is going to react with the residue over here, then there will be reaction, or there will be separation of this N terminal from this whole chain by selective detachment. Selective detachment of N terminal residue by anhydrous acid. By anhydrous acid. This is going to occur. Now the derivatives are then isomerized to give the substitute, which is called as phenyl thiohydrantoin, and the isomer, that is the phenyl thiohydrantoin, which can be washed and identified by chromatography. And the cycle can be repeated till the efficiency which is reached is 98%, which tells us almost 50 amino acid sequence in the one go, right? So the last thing that is needed over here in this case is the derivative that is being obtained, and the derivative to give a substituted phenyl thiohydrantoin. So the derivative, so the derivative then isomerizes to give a substituted phenyl thiohydrantoin. This is obtained, and which can be washed off and can be identified by chromatography. Washed and identified by chromatography. This is the thing. So efficiency is approximately 98%, and 50 amino acid can be done in a single go. Now talking about protein sequencer, that is a further elaboration of Edmonds method only, that is the automated thing. So sequenator, protein sequenator. So a protein sequenator is basically a machine that performs the Edmonds degradation in an automated manner. So a sample of the uh, protein or a polypeptide, it is immobilized, and in the reaction vessel of the protein sequenator, and the Edmond degradation is performed. So each of the cycle is going to tell us about one amino acid from the protein, and the cycle would be kept on going on, and then finally the determination would be done by the HPLC method. So in the protein sequenator, that another thing which is different than the manually operated Edmond reaction is use of HPLC. Now talking about some of the limitations of Edmonds reagent, limitations of Edmonds reaction. So because the Edmonds degradation proceeds from N terminal of the protein, it is not going to work if the N terminal is chemically modified. So that can be one of the issue when we are going for Edmonds technique. Then sequencing will stop if non-alpha amino acid is encountered. Since the favored 5-membered ring intermediate is unable to be formed, which we have seen, right? So that won't be formed if there is absence of alpha amino acid. Then Edmond degradation is generally not at all useful to determine the position of disulfide bond, and it requires the peptide in the very small amount, that is 1 picomole, or in even lesser than that, right? So these are the steps which are involved in the protein sequence determination. Then another thing, which is the gold standard for identifying the protein sequence in the extremely quick and accurate manner, is mass spectroscopy. So we are not including mass spectroscopy over here, because mass spectroscopy needs a whole different lecture to be covered into. So mass spectroscopy will be talking about it in detail in further some other class. So like this, we have completed the process of protein sequence determination by Sanger's method and by Edmonds method. Sanger's method, the Sanger's reagent is fluorodinitrobenzene, and the Edmonds reagent is uh, PITC, that is phenyl isothiocyanate. And rest all the steps we have seen that what needs to be done for performing both of the methods. So like this, we have completed the topic protein sequencing, and as we say in the end, let's crack CSIR UGC NET and GATE 2021 together. And that is the name of our channel as well. So if you haven't subscribed our channel till now, subscribe the channel, hit the bell icon for getting all the notifications of the upcoming classes. If you really like the video, give it a thumbs up. Really motivates the educators like us. Recently a major discount is going on in the plus subscription. All the topics and everything is there in the plus subscription. So do not forget to use my referral code FB01 for this plus subscription. And even for special classes, you will need my referral code FB01. So do not forget to use my referral code FB01 when you are going to download the app and go for the special class. And in the plus subscription, my referral code is going to get you a 10% additional discount. So this is it for now. Thank you very much for your patient listening and for coming to the class. And I hope to see you everyone at 5 o'clock in the 3 o'clock in the noon, and tomorrow 10:30 a.m. in the morning for CSI class, and 3 p.m. in the noon for GATE class. So this is it for now. We'll be meeting again tomorrow. I hope you people have understood the thing. If you haven't understood the thing, you can write down in the comment box. Whenever I'll be going through my video, I'll try to solve your doubts and queries. I'll try to take up your question. And if you want any specific topic to be explained by me, you can write down that too as well in the comment box. So this is it for now. We'll be meeting at 3 p.m. today and tomorrow 10:30 a.m. in the morning, and we'll study some other interesting topics. So this is it for now. It's a bye from my side.